So I work on the integrated control of false codling moth on stone fruit and table grapes. But before we can speak about the control of any insect pest, we first have to understand its biology and therefore its life cycle. So to illustrate this for false codling moth, I teamed up with an animator, and if you maybe just get the lights, we made this short clip on the life cycle of false codling moth on table grapes, but it's the same for stone fruit. So the female moth lays its egg on the outside of the fruit, and the first instar bores into the fruit, after which the fifth instar emerges. It drops to the soil, and here it will gather soil and silk around it so as to form a pupae within the first 2 days. The pupae will then mature under the soil surface for about 2 weeks, after which the adult moth will emerge. The adult moth can then move into new environments, perhaps an orchard next door, and at about 2 days of age will become sexually mature and go in search of a mate. After a mate is found, the whole process starts all over again. So now that we've got a good understanding of how the life cycle works, we can speak about the control. I suggest a control at each stage of the life cycle. For the adult stage, we can use mating disruption, which is the broadcasting of the female pheromone throughout the landscape so as to decrease mating events and thus suppress the population. And then for the other stages, the immature stages, I suggest entomopathogens or insect killers, such as nematodes and fungi. So straight to the results. From our experiments, we found that as little as a third of the recommended field dose, that was for citrus recommended field dose, in stone fruit and table grapes, we can get up to 85% disruption, and this does not increase up until the field dose. However, if we increase the field dose by 1.3 times, we can increase it to 96% disruption. For the egg stage, we found the first record of virulence against FCM eggs, and this is a really good finding as we can stop the insect or kill it before it makes damage or causes damage to the fruit. Some of our strains causing more than 50% mortality. The pupal stage was the least susceptible of the FCM stages, with our best EPN species causing only 30% mortality. More work should focus on this stage, and fungal or fungi has great potential in this for this stage. However, larvae was the most susceptible in the lab stage, with some species causing 100% in every single trial I did. As it was the most susceptible stage, I also tested all my collected fungi against this stage, with some strains causing more than 75% mortality in the lab. However, the EPNs still proved to be the most successful against the larvae, and thus field trials were done on it. The species caused about 70% mortality in field and persisted for more than 4 weeks. It is therefore clear that we can control false codling moth even if they hide. Thank you.